Now, in chapter 15, we dug into the regulation of gene expression, but primarily in prokaryotes. In this chapter, we're going to touch on some of the differences that we know about that exist in eukaryotes. So, there's a lot more opportunity and many more mechanisms that contribute to the regulation of gene expression in eukaryotes. And this is where we left off in chapter 15, and so we'll jump right in. And the first is I'm just going to give you a high level overview of ways in which gene expression might be regulated in eukaryotes. And for most of these, it's going to be obvious why this is not a possibility within prokaryotes. So the first is the regulation of chromatin remodeling. Okay, again, this is a high level overview. I'm not going to dig into specifics right here in these next couple slides. The remodeling of chromatin is, of course, unique and specific to eukaryotes and their chromatin. But as you see, this finest level of chromatin structure, right, these nucleosomes, in which roughly 150 nucleotides is wrapped around a histone octamer with a spacer histone in between the nucleosomes. This is a level of compaction that we perceive of in our discussion of DNA compaction as being relatively little in the way of compression or compaction. But this DNA is wound around these histone octamers and is not terribly accessible for polymerases for the process of transcription. And so cells are able to regulate the extent to which this double-stranded DNA molecule, here looking like a pink piece of spaghetti wrapped around these histone octamers, how accessible that double-stranded DNA is, or whether it's all wrapped up around these histones. Okay? Now, there is also regulation at the level of transcription, right? So is mRNA made or not, right? And we talked a lot about that in prokaryotes, so this isn't terribly different, but the regulation of whether RNA polymerase comes in and makes a pre-mRNA or a transcript of a particular coding region, that is also one of the levels of regulation we observe in eukaryotes. And then the next step we know that occurs within eukaryotes is the processing of this pre-mRNA, which consists of adding the 5' cap, the poly A tail to the 3' end, and then also removing all of the exons. Okay? Each of these is quite unique to eukaryotes, and whether or not it is done affects whether or not there is a significant amount of this gene product produced by translation. Okay, so let's say we do have messenger RNA that is created through transcription. That pre-mRNA is processed, so it's spliced and capped and polyadenylated on the 3' end. The next step is that RNA molecule, that mRNA, needs to leave the nucleus to where the ribosomes are. We all know now that the ribosomes are where, or the means by which these messenger RNA molecules can be translated. And so they have to be in the same place as the RNA molecule. Ribosomes don't go into the nucleus. The mRNA has to come to them. Okay? And the movement of that messenger RNA molecule out of the nucleus is actually regulated. Okay, it's not just a passive diffusion of random RNA molecules. They are carried out of the nucleus, and if the cell is not needing to express these genes at this point, then it will not transmit them across the nuclear membrane. Okay, and all of these incremental or different checkpoints in which regulation is possible are in part due to the need to change the mind, basically, of the cell. Once it has, quote unquote, decided to express a gene, right, there's a need for that gene product. it will initiate transcription and then translation. But sometime between the cell learning of the need and the translation, the situation might change and the gene product might no longer be needed. Okay? Here, that regulation can take the place or take the form of the failure to transport that messenger RNA out of the nucleus. Okay? Certainly, once the ribosomes interact with the messenger RNA molecule, we think that they would begin translating, and that is generally the case, although it's also possible that ribosomes are prevented from interacting with the messenger RNA molecule. Once that messenger RNA molecule gets out of the nucleus, there are a number of things that could happen to it. Okay, and one of which is that the messenger RNA could be degraded. I've spoken to you about how messenger RNA and RNA in general that is single-stranded is quite fragile and subject to degradation. Okay, for that reason, we add that 5' cap and poly A tail to the messenger RNA molecule when it's processed. But there's still a chance that the messenger RNA could be degraded, and that's one of the last things we'll touch on in this section, actually how that happens. Now, the next step after transcription and moving that messenger RNA molecule that has been processed out of the nucleus, getting into the ribosomes, you start translating, okay? Whether or not translation happens and the extent to which it happens for any particular type of messenger RNA can be regulated, as can be the modifications of the polypeptide once translation is complete. Remember, translation produces simply a string of amino acids, this polypeptide, that in order to become functional must be properly configured, potentially interacting with other molecules, other polypeptides, and other proteins in order to be a functional molecule. Whether or not that happens, those modifications and foldings and pairings and groupings, whether or not those happen, okay, is also a mechanism whereby eukaryotic organisms can regulate the expression of a gene. All right, so now we're going to start back at the beginning there. I gave you the big overview. We're going to start and hit on a couple of these in a good bit more detail so you have a more nuanced understanding of the ways in which expression can be regulated in eukaryotes. Okay, so the first thing I want you to think about is the accessibility of that information in the genetic code. Yes, it's hidden away in the nucleus, but that's not necessarily a problem for the cell to access its own information. But remember, when it is compacted into that chromatin form, it can be a bit difficult to access. All right? Now, when these chromosomes are in this relaxed chromosome state, the analogy that I used before of this tangled ball of yarn, the nucleus isn't quite like that. Each chromosome, even though it's kind of in this relaxed chromatin state, has its own chromosome territory. Okay, so you can see here within the nucleus, each chromosome has been dyed a different color. So all the DNA on a single double-stranded molecule of DNA has received its own color. And in looking at this nucleus, you can see that there are distinct regions of red and blue and green and purple. And that is because each of these molecules of DNA is occupying, in its own relaxed way, a very distinct space. And there's very little overlap or interaction with other chromosomes. That makes it much easier for the process of transcription to occur when it is easy to identify and find particular regions of a chromosome. And between these chromosomes are what are known as interchromosomal domains. You can just think of that as the personal space of the chromosome that facilitates the access of the genetic material within those chromosomes. Okay. Now, there are two mechanisms whereby the structure of DNA is chemically altered, and that affects the accessibility of the information contained within the molecule of nucleic acid for transcription. The first of these is chromatin remodeling. And what is meant by chromatin remodeling is shifting the way in which a nucleic acid strand wraps around and interacts with those histone octamers and spacer histones. Okay? If you can create an area where the DNA is actually exposed and not tied up with all of these histones, that makes it much easier to transcribe the information contained within it. Okay? If that DNA is tightly packed and wrapped around histones, it's fairly unavailable for transcription. So the chromatin must be remodeled for a particular region of the chromosome if it is to be transcribed. The other thing I want to touch on is one that we don't speak about too much in genetics. It's probably the only place you're going to hear about it, but it's not a huge part of the material that we cover, and that is methylation. Methylation is a critically important mechanism for the regulation of gene expression. And one of the more interesting discoveries in the past few decades is that these patterns of methylation can be heritable, okay? And they're accumulated during your lifetime. And so let me talk a bit about what methylation is. It is the addition of a methyl group to the bases and sugars of the DNA. And what it does is it inhibits the expression of a particular gene region. So here you can see up at the top that at the finest scale of this double helix, that's actually not even twisted into a helix just yet, you can see all of these methyl groups that are added, okay? And what these methyl groups do is they're attached to the base of the nucleic acid, and they poke them, that methyl group sticks into the major groove of that double helix. It kind of hangs out in that area, which is an area that is a large groove because it is one where polymerases need to interact with the DNA. If that polymerase molecule can't get in there because there's a methyl group stuck there in that major groove, then it reduces the expression of the gene around it. Okay? Now, what's been so interesting about DNA methylation is that researchers have learned that as you expose yourself to things, whether it's a period of starvation or drug use or high stress, that can lead to methylation of certain parts of your genome, presumably to reduce expression of certain things. But what's fascinating is that this is not necessarily methylation to your germ cells or your germ cells only. This might be expression changes that take place across your entire body, but the effects can be seen in your offspring because they can inherit your same methylation patterns. Pretty amazing. So if we look here at a somewhat more informative image of methylation that I pulled from the internet, you can see at the top the difference between methylated and unmethylated DNA, okay? So it's primarily on these cytosine residues that you will get these methyl groups attached, right? And the difference between the two DNA strands in the middle, the one with the little green squiggles off the histones, suggesting that those histones are acetylated. So that molecule of DNA wrapped around histones, that gene is basically on or available for transcription. The chromatin's relatively open, there's no methyl group on the cytosines, and the histones are acetylated. If, on the other hand, you see something more like the condition on the bottom there of the middle, where you have the cytosine residues being methylated, the histones are deacetylated, this is highly unlikely to be expressed. This gene is basically turned off. It's covered up, it's hidden, It's not usable. Okay? So methylation and deacetylation of nucleic acids in our genome, those also contribute a great deal to the expression of the genes around them. Now, when we spoke of the prokaryotic gene regulation systems, much like this, this is the lac operon below, we spoke about promoter regions and how those affect the regulation of the expression, the levels of transcription and translation of a particular gene. Okay? So those promoters were hugely important in the regulation of gene expression in the lac operon. Well, promoter regions are also incredibly important in regulation of genes within eukaryotic cells. And not only these promoter regions, which are the regions where the polymerase will bind and initiate transcription, we also have other regions that are cis-regulatory elements of these genes that can be characterized as either silencers or enhancers, with silencers acting to reduce gene expression and enhancers acting to promote gene expression or enhance or increase the amount of expression of a particular gene. Notably, these are cis-regulatory elements that are generally somewhat separated from promoters. They're not so distant as a gene that might code for a repressor protein, as we saw in the lac operon, but they are somewhat distant from the promoter region of the gene, which is necessarily adjacent to the gene. Now, these cis-regulatory elements might consist of things that I've already mentioned, like TATA boxes, CAT boxes, and GC boxes, all three of which are illustrated here, and an example from your text where the levels of expression of a particular gene region downstream off the screen to our right, the levels of expression of the gene back there are quantified if there is a mutation to any one nucleotide in the region of the chromosome that we're looking at. So from left to right, in that center graph, anytime you see a bar, a vertical bar sticking up in that graph, that bar is telling you if there is a mutation to that specific spot, okay? And notice that it could be anywhere. It could be in just the gray before the promoter, which is off to our right and before the gene, or it could be to the CAT box, GC box, TATA box, etc. So what do we see happen to levels of expression when we have mutations to regions that are outside of the gene, that are not in the promoter? Okay. Well, if you look just above that little red arrow to the right where initiation begins, you'll see that most of the bars in that region that rise up from the bottom of the graph rise up to a level that is approximately 1.0 on the y-axis. Look over to the left, see where 1.0 is. What that is telling you is when a bar is right about 1.0, that is relative transcription level. What it's telling you is how much transcription of the gene to our right do we get if you mutate this spot, okay? If it's less, you're gonna get relative levels of transcription that are less than 1.0, right? Let's say you make 50 molecules instead of 100, then the relative transcription would be 0.5. If you make twice as much, then the relative transcription might be 2.0, okay? So let's work our way from right to left. Now, the first thing I'm going to note here is that there's some black dots on the bottom, basically our x-axis. And where those black dots are, there is no vertical line extending up from them. So what happens when you mutate those nucleotides? Because each of these spots, whether it's a dot or a line, indicates a single nucleotide, okay? So in these areas where there's a black dot, no vertical line, what does that tell you about expression of the gene to our right if you mutate that nucleotide? the relative transcription level is 0, meaning you're not transcribing at all. A mutation to some of those points is effectively completely inactivating the gene downstream. Okay? Now, most of those vertical bars that stick up from the x-axis, they're coming in right at the level of 1.0, plus or minus a wee bit. Okay? So mutations to most of those spots don't really matter. Now, when we get to the TATA box or the CAT box or the GC box, you'll notice a very different story appear. Every nucleotide in the TATA box is important. If you mutate it, if you change it from a T or an A to something else, that relative expression level drops to 25 to 50%. So you lose a lot of expression if you mutate one of these cis-regulatory regions. Okay? Now, I want you to note here that the GC box is longer than just GC. The TATA box is 5 nucleotides long, not 4. Okay? These are just names that are applied to give you an idea of what the repeat is in this regulatory region. So the TATA box might end with 2 A's or have 2 A's in the middle, but it's TA rich. Same is true of the CAT box and the GC box. Okay, the GC box are just going to have GC, GC, GC a few times. So you'll notice here that most of the mutations don't have any effect. Most of these bars are at 1.0. The ones in these particularly noteworthy regulatory regions certainly reduce gene expression, and there's only, it looks like, 2 mutations just before the CAT box that result in much higher gene expression levels, which is also quite interesting. Okay, so genes can have lots of different cis-acting regulatory elements like this, okay? And here you can see a particular gene that is, well, this cis-regulatory region for a particular gene that was profiled by your text. And in this, you can see not only that there are lots of different types of regulatory regions, but that there are also many different transcription factors, those little balls and ovals beneath the DNA strand. There are many different transcription factors that interact with many different regulatory regions to affect levels of gene expression, okay? And so this is how your gene expression can be more like a faucet than a light switch, okay? Expression of genes is not on or off, okay? Expression of genes can be highly regulated to a very fine level, especially when there are so many different regulatory elements or transcription factors that can fine-tune the amount of the protein that is produced through gene expression, okay? So let's just take a peek at an example of how these transcription factors enable transcription by RNA polymerase II, which in eukaryotes is what we use to transcribe gene regions, okay? So you'll see that we've got a double-stranded DNA molecule here. We've got the TATA box identified. And what comes in and binds there are these first 2 transcription factors that are then joined by others, 2A and 2B, okay? So these first 2 proteins come in and then interact with 2A and 2B. What this is then followed with is the bringing in of RNA polymerase II. It's carried there by transcription factor 2F. You get the polymerase II there, and then these other transcription factors start dropping in, 2E, 2H, 2J. All of these together create a highly conductive environment for the transcription of a gene region, okay? And so the last thing I want to point out with respect to these molecules that interact with the DNA itself and contribute to gene expression. Remember when I showed you that schematic that indicated that these silencers or enhancers might occur quite a ways upstream from the gene? Well, they interact with molecules that may contribute to whether or not this complex formed by the polymerase II and all of these transcription factors really takes off. If these activator proteins that have interacted with the enhancer region complete their assembly, the whole DNA molecule can fold back upon itself so that these activator proteins are held together in a particular way. They're a certain distance from the promoter region. They bend over and interact directly with this transcription complex that has formed with the DNA or RNA polymerase II. So all of these molecules interacting with these regulatory sites in the genome contribute to gene expression. Okay, so I want you to think back to a couple things we've talked about with respect to the organization of the genome. Remember how we talked about some mutations, lots of mutations to the genome have no effect. Sure, that's true, but are the only ones that have any effect within the codons of the gene? Clearly not. Clearly these regions of the genome, Tata box, enhancers, cat boxes, all of these things, if you're to have mutations there, those will have an effect on the phenotype, okay? Through their contribution or the lack thereof with respect to the regulation of expression of a gene that they contribute to controlling. Okay? Now, let's assume we go ahead and get a transcript. We get a messenger RNA molecule. Okay? One of the things that is touched on in more depth here than in previous chapters where I did introduce it is alternate splicing. Okay? So the processing of these pre-mRNA molecules, as you know, includes the attaching of the 5' cap, the poly A tail to the 3' end, but also getting rid of all those exons. But what if you don't get rid of all the exons in the same way every single time? Your text has a very large figure that goes through all of these possibilities. You don't need to memorize the terms on the left here, the different types of alternative splicing. Oh, and that reminds me, you don't need to memorize all of the different promoter regions and transcription factors that bind in the previous slides. Okay, so all those transacting molecules that you saw, you don't need to memorize all of those. I just want you to understand how complex the regulation of transcription is in eukaryotes. So here you can see the way in which certain exons can be included or removed in the processing of messenger RNAs. So in the first example of cassette exons, you can see that there are two possible mature messenger RNAs to result from the expression of this particular gene, one of which does not include exon number 2, the darker brown one in the middle, which is quite surprising. You can have alternative splice sites in which the splicing out of introns, you can process this pre-mRNA on the left in two different ways, one of which excludes a portion of the exon, colored blue here, okay? And all of these, the introns, are that kind of thicker brown bar that connects the exons, which are much thicker. Okay, in intron retention, sometimes introns are not removed, and that would certainly affect the expression of this messenger RNA molecule. Mutually exclusive exons, so you might end up with two different mRNAs, each of which contains a unique exon that is not present in the other, so they're mutually exclusive. You can have alternate promoters. Okay, so where translation starts at two different regions. or you can have alternate polyadenylation. So the poly A tail can be added in different spots, either after the second exon or it can be after the third exon. Okay. So there's one example in your text where they go into much more detail about the specifics of the alternative splicing of this particular gene. So this gene is called the calcitonin gene-related peptide, okay? And when it's transcribed, there are 6 exons, okay, and a number of introns. But in thyroid cells, when this gene is expressed, there's a poly A tail added to the end of the 3' end of the 4th exon, okay? So you throw that poly A tail on there as you are transcribing, and you don't get further transcription of exon 5 or exon 6, okay? So what you end up with is what is above in the middle of this image. So I want you to start at the bottom and work your way up with this. In the middle, we have a CT mRNA that is how this gene is expressed in thyroid cells. And this results in circulating calcitonin peptides. So it's a hormone that circulates in your blood and regulates calcium levels. Okay, so your thyroid regulates that. You might know of some older folks. This is in my family's, mine and my wife's. This is quite common in the women in the family where they have their thyroids removed, they're having hormonal issues, and after that they have to take medication in part to ensure that they are properly assimilating calcium into their body and not losing calcium. So this is an important gene, and in thyroid cells it's expressed in this way, the messenger RNA including only 4 exons, creating this calcitonin peptide. Now in neurons, when this gene is transcribed, it skips over the 4th exon and polyadenylates the 6th exon. So what you have is a messenger RNA with exons 1, 2, 3, 5, and 6 that is translated, and the gene product is this GCRP, okay? And this stimulates dilation of blood vessels. Okay, so doing 2 very different things, calcitonin and GCRP, are doing very different things for the body. And so this difference in function is entirely due to the way in which these genes are transcribed and translated in very tissue-specific ways. Now, alternative splicing is the answer to why we have so many proteins that we can make and so few genes in our genome comparatively, okay? So you have about 25,000 genes. 30 to 60% of those can undergo alternative splicing. I know that's a wide range, but just realize that it's a large proportion of your genes can undergo alternative splicing. So the interesting thing is, despite the fact you have 25,000 genes, you can produce several 100,000 different proteins. And this is clearly through the ways in which these genes can be alternatively spliced. Here we have an example that's described in your text. This is figure 1610, and this is a Drosophila or a fruit fly gene, DSCAM. And what is remarkable is that this gene encodes a protein that regulates the growth of axons, so nerve cells during development. And the messenger RNA, the pre-messenger RNA, when it is processed, it will be processed so that it will only contain 1 of the 12 possible exons for exon 4. So you'll see 12 little red lines here under exon 4. Each mRNA gets only 1 of those 12, okay? It only gets 1 of the 48 possible exons for exon 6, all those blue lines. It gets 1 of the 33 possible exon 9s, and 1 of the 2 possibilities for exon 17, okay? All of these possibilities, you've got one of 12, one of 48, one of 33, and one of 2. Okay, this is a plethora of opportunities for unique combinations. And indeed, it turns out there are over 38,000 unique possible combinations that can be created with this pre-mRNA. Okay. Now, what's even more remarkable is that researchers have studied to see whether these fruit flies actually need all these different possible combinations, or if they only use one or two of them. And it turns out that it does appear that they need to be able to produce all of these different varieties. Okay, so alternative splicing is a great means of regulating gene expression. Now, the next one I'm going to talk to you about is, let's assume that we have gone ahead and transcribed a gene and made a pre-mRNA. Okay, that's the first hurdle. Then that pre-mRNA is processed. 5' cap, poly A tail, exons gone. We just talked about a number of ways in which the selective removal of exons can have different results. Now we move on to, we have a messenger RNA molecule. Let's say we did take it out of the nucleus. It's ready to be translated, but if we don't need it anymore, how do we get rid of those messenger RNA molecules? So I've told you that we have to add that 5' cap and poly A tail because these single-stranded nucleic acids are susceptible to degradation. And the abundance of any messenger RNA molecule in our cells at any one time is the product of two factors. How much of it are we making and how fast? And then how much is being destroyed and how fast is it being destroyed? Okay, so if we made a bunch of it and we don't need it anymore, then degradation happens. And there are a couple of things that work to degrade these messenger RNA molecules. The first is deadenylase, which, as it looks like, kind of Pac-Mans, or chews down this poly A tail. Okay, so it will chew down the poly A tail from the right, taking it down from hundreds of A's in a row to tens or fewer. Okay, so you see in this example, we're going from having 200 A's hanging off the end, that protective bumper on the 3' end, to somewhere in the neighborhood of 10 of them left. Okay, so the vast majority of this poly A tail has been destroyed. And if you continue down to the bottom right, you'll see that when this deadenylase tears up and destroys this poly A tail, then an exosome may form and decay or destroy, eat up this mRNA from the 3' end. Okay. The other thing that can also happen is that 5' cap, it can be decapped. There's an enzyme that comes in and will remove that methylguanosine cap from the 5' end. Okay. Once it's decapped, a molecule called XRN1 can come in and chew it up from the 5' end. Okay. So between the exosome and XRN1, these messenger RNA molecules can be chewed up from both ends if the cap and the tail have been removed, or the tail have been removed. Okay, now the last thing I want to mention to you is that messenger RNA that is produced, gets out of the nucleus, might be held in check or kept hidden effectively unless it's in the right place. So we think of cells as being very small and having not much room, but realize that we're at a very different scale than a cell and all of the things within it, all of the molecules that we are talking about. And recognize some cells are much larger than you think, okay, than you traditionally think about. So this is an example described in your text whereby the regulation of actin messenger RNA is controlled within nerve cells, okay? And so in this cell, this gene is transcribed, but it is immediately bound by this ZBP1 molecule. which escorts it out of the nucleus. But because it is the molecule that carries it out of the nucleus, as soon as that thing emerges from the nucleus, where ribosomes are ready to read it, ZBP1 is also hiding it or preventing it from interacting with ribosomes. So you're not going to get any translation. ZBP1 interacts with another molecule, MP, which is connected to microtubules associated with actin, and it literally acts as a monorail or a train that takes this gene transcript all the way down to where it is needed, which is down near the actin filaments. Okay? Once it reaches that area, there is a locally abundant molecule, SRC, that will interact only with ZBP1. When it does that, it phosphorylates it, knocking ZBP1 off of the messenger RNA molecule, allowing a ribosome to form and translate this messenger RNA molecule. Okay? So this is an instance where we're seeing a few things, the first of which is that there are molecules that can bind to a messenger RNA, like ZBP1, that are not destroying it, but they're hiding it and keeping it from being translated until it's in the right spot. How does it know it's in the right spot? When the SRC molecule comes in and removes it, because the SRC is only near the actin. We need this gene translated near the actin. Boom, everything works out perfectly. Okay, so there are some bits in this chapter that I did not go over, so like long non-coding RNAs and post-transcriptional regulation. I didn't really talk much about these non-coding RNAs in 16.8. Just be sure you focus on the mechanisms that I've described and understand ways in which gene expression can be regulated. Okay?